Okay. Karinne, take it away.
All right. Hi, everyone. My name is Karinne Tenenbaum, and I'm a third-year student at Yale University in the United States, majoring in ecology and evolutionary biology. And I'm excited to present Bite Size, the use of model prey to measure predation pressure of insect herbivores during the secondary succession of tropical forests. So the presentation's in English, and the slides are in Spanish, but there is a QR code on the first slide and the last slide if you would like an English translation of the slides. And this is the project that I conducted during these past 2 months under the supervision of Anita Weisblog and Liza Comida as a fellow of the Yale School of the Environment Tropical Resources Institute. And at this time, I'm still collecting data, so today I'm going to talk about the idea, the methods, and a little bit of preliminary results. Tropical rainforests have experienced extensive human exploitation over the last century, from timber extraction to wildfire to agricultural and residential development. For example, in the canal area of Panama, many forests have been clear-cut in the past for small-scale agriculture use and pineapple plantations. More than 70% of the world's natural forests have been severely degraded and are now in some stage of recovery. And a natural recovery, in which a forest is growing back on degraded land, is called secondary succession. Long-term conservation of tropical forest biodiversity relies on maintaining rich species diversity in these dramatically altered landscapes. And this crucial species richness is known to be regulated by species interactions, such as those between plants and their herbivores. However, less is known about the effects of higher trophic levels, such as predators of insect herbivores on primary producers, which may ultimately impact patterns of forest recovery and explain their high unpredictability. So in this study, we were particularly interested in examining how the predation of insect herbivores varies along secondary succession. Insect herbivores, such as caterpillars, are the most crucial primary consumer in tropical forests. They cause tropical trees to lose nearly 1/4 of their foliage. And higher herbivore density has been shown to lead to a decrease in plant biomass, suggesting that insect and vertebrate communities influence regeneration dynamics in tropical forests. And so the predation of these herbivores across trophic levels can induce significant ecological and evolutionary consequences, effectively limiting herbivory and having cascading effects on plant performance and community composition. And so predation pressure is an integral part of the secondary succession puzzle, and yet insect predation is difficult to observe live, since predation often leaves no trace, so there's very little prey remaining. The event is happening very quickly. The herbivores themselves are small, agile, they have nocturnal eating habits, and so some methods to measure predation pressure often use live prey, such as mealworms, to attract natural enemies. And unfortunately, these techniques prove challenging themselves, since you either need to watch the prey for an extended period of time, or you need to invest in a really expensive camera, not alone having to deal with the ethics of using live prey to attract natural enemies. And so the use of artificial plasticine caterpillars, I want you to imagine Play-Doh in your head, with fast-setting glue, has become an increasingly popular and successful method used in local, regional, and global scale studies to assess predation pressure on insect herbivores that is both inexpensive and convenient. And the plasticine is non-toxic and odorless and attempts to mimic undefended geometric caterpillars. It's available in different colors and sizes, so you can look at specialist predation, and the malleability of the material itself allows you to visualize the evidence of attack with visible marks left in the clay. And I actually brought some of these caterpillars here for you all to have some first-hand experience. There's not enough for everyone, but you can share like you did once in school. And I encourage you to take a caterpillar and you can put your fingernail in it just to see what the mark leaves, which is what we accidentally did numerous times in the field. And then you can try and smooth it over. You can mark it, but please don't bite it. And whoever is the best at smoothing it, you're welcome to come join the team. Research on secondary succession of forests post-disturbance is best conducted through direct observation over a long period of time, hundreds of years, which, as you can expect, tends to be a difficult endeavor. And so the studies of secondary succession aim to use closely adjacent forest stands in similar habitats with similar histories of disturbance, and we say that the key assumption to being able to do this is that it's fairly safe to expect that more recently disturbed stands will eventually resemble these older stands. And so in this way, the chronosequence affords tropical forest researchers the ability to investigate the temporal aspect within their study areas. And over the last 8 weeks, I was fortunate to work in an already established secondary forest chronosequence comprising 10 different forest sites of 5 successional ages: 9 years, 49, 69, 99, and 129 years. All of the sites are within the Barro National Monument, 3 of which are on BCI. And for all of these sites, there's multi-decade forest recovery data available thanks to there's existing vegetation plots and ongoing research by D.C. Dent and Liza Komita. And so in early successional stages, abiotic factors can greatly influence insect herbivory through higher air temperature and lower humidity, and these environmental conditions likely also act on the behavior of natural enemies. And this plant species richness increases with successional age, so this allows for a coexistence of a larger diversity of specialist herbivores, as well as their predators. And so the entire network of coexisting species becomes progressively more complex. All of the forest sites have a 1 hectare observational vegetation plot. And around the perimeter of the plot, we set up 5 subplots, roughly 5 by 5 meters, spaced at least 50 meters apart. And in each subplot, we randomly marked 20 seedlings, and we marked it in a way so that it wouldn't affect the ascent of the natural enemy coming to attack our caterpillar. So for example, if we place the caterpillar all the way on the right of this plant, we place the tagging on the left. So with 5 subplots per forest site, and we had 20 seedlings, and we had 1 caterpillar on 1 leaf on that plant, we set out 100 caterpillars in each site. And since leaf position and leaf characteristics may affect the visibility and accessibility of the prey, we chose to use leaves that were in the top third of the plant, 40 to 100 centimeters from the forest floor. And please note the poles were just to show some scale, they weren't actually there while we were doing the study. And for standardization purposes, we only chose adult green leaves that were simple, completely expanded. They were about medium size, all healthy, with little to no herbivory, no serrated edges, and they were all smooth. So they didn't have hair or trichomes. And then we would place the plasticine caterpillar on that leaf along the midline vein for maximum stability, attached with just a little bit of glue, and we came back after 48 hours to see if there were any marks or visible evidence of attack. And so in total, we set out 1,000 caterpillars to test for variation in predation pressure along the secondary succession gradient. And we hypothesized that predation pressure of the insect herbivores increases with increasing successional age. And that's due to this buildup of plant herbivore predator networks. And so we predicted higher predation pressure and higher diversity of predators in older sites. So to start off, I made 1 caterpillar. And then I made 1,000 caterpillars. And then 1 by 1, 100 by 100, we went out to the sites and we placed all of these caterpillars out. And unfortunately, due to some inclement weather here in Panama, we had some fall off the leaf. And we actually had a lot of them fall off the leaf. And you may expect that this was problematic because even if we found marks in the caterpillars themselves, we had no way of knowing if that was due to an attack that happened while that caterpillar was on the leaf or when it was already in the soil, which is a totally different system. So we needed another way to keep the caterpillars on the leaf. And after some prototyping in the lab, where we basically put the caterpillar on the leaf and we held it under the faucet, and I told myself that if it can withstand the faucet, it can withstand Panama weather. And luckily we were able to find a method that involved pins. And so essentially we threaded the needle through the clay and into the leaf. And even though there's only 3 points of contact, it held up under the faucet, and more importantly, it held up outside in the field. So I'm happy to report that of the 800 caterpillars that we set out, only 48, about 6%, fell off, which was great. And not only did they stay, they allowed us to visualize a part of attack that we weren't able to see before with the glue. We could actually see when a natural enemy was trying to remove the caterpillar from the leaf itself. And they stayed, and they were devoured. And so we went out after 48 hours and we collected all the caterpillars that we had, but only the ones that had visual attack marks. We used a little magnifying glass out in the field. We kept intact and we saved for later analysis. And so all of these caterpillars are going to be photographed with a stereoscope. And the idea is that we're able to look at the mark and attribute it to a specific natural enemy. So we have some ideas that scattered bite marks are associated with chewing arthropods. Does anyone want to guess what this top left one is? Just shout it. Bird is correct. You're looking for like a deep triangular mark, not necessarily oscillated and bird, but who knows? And so we have pretty good inclination that those are bird marks. And also if we had a raised one, you could usually find bird marks specifically on those. But we don't know which arthropod did this, we don't know which arthropod did that, and so enter bite guide. we have a goal of trying to match this attack mark to the attacker itself, and so we decided to make a comprehensive catalog of different arthropods and their bites so that we can have this side-by-side comparison. And so we collected insects on BCI, and we put them in a small container with some clay for 24 hours to see whether they would bite it and what kind of bites they would leave. We were actually lucky enough to observe a live predation in the field, so when we came back to do the 40-hour check, this pill bug was actually on our caterpillar, and so we can see that this was directly causing this specific mark. And the catadid was our first guinea pig, if you will, and we saw that it left these longer linear trends. And now it is your turn. So I invite you to play a little bit of Guess Who, and see if you can match the attacker to the attack mark. Take 10 seconds, turn to your neighbor, and see if you can match it. It's okay, you won't get a grade. All right, do we have some ideas? Volunteer? All right, what do you got?
I'm going to guess that the salad did the middle one.
Okay.
Yeah.
Any counters? That's what everyone thinks?
Yeah.
Okay, here we go. So we have, for kinds of the cricket, grasshopper, large. So that's the cricket. That's the grasshopper. I gave you guys a little bit of hint. The last one that the catadid was a little bit similar. And then that's the beetle. So not necessarily intuitive, but that's why we have the bite guide. And now for some general results. So here are some preliminary data. Along the x-axis, you'll see forest age, and on the y-axis, you'll see the percentage of caterpillars attacked. Note that we have 8 sites here. We have 2 additional sites. We did an additional 99 sites this morning, and I can tell you that it looks very similar to that one. And we're going to do one more site in our 9-year-old plot as well. And so we measured predation as the percentage of attacked caterpillars per subplot. So for example, we know that there's 5 subplots per area, and each one has 20 caterpillars. And so for example, here at EcoForest 2, we had a range from 0% to 20% of the 5 subplots that were there, right? This one has only 1 site because we're going to do the second one, but all the way on the right, for the 129-year-old sites, you saw that for the 5, for each of them, you have a range from, for example, the red is from 0% to 15%, and the blue is from roughly 15% to 26%. Excuse me. We see the predation range is from 0 to 30%, which is pretty comparable to previous studies. And we see this slight peak at the 49-year-old sites and the 129-year-old sites, which is in contrast to our hypothesis that we were expecting greater predation pressure with an increasing successional age. And so we used a generalized linear mixed effects model, analyzing the proportion of attacked caterpillars over forest age, controlling for forest site and subplot, and performed post hoc Tukey tests to test for differences. And all the p-values were larger than 0.05. So even though we see these trends, there isn't a statistical difference among the groups. We also looked a little bit at diversity. So right now we're checking between birds and arthropods, and so we see an increase in the bird attacks, specifically in one 49-year-old site. There was an additional bird attack in the 99-year-old site this morning, but this is interesting because this suggests that there may be some correlation between predation pressure and greater predator diversity. So as of now, our preliminary results don't suggest that the age of the forest has a significant effect on the predation pressure across the chronosequence, but perhaps that specific site variation has a larger effect. And what's interesting that our higher bird peak at the 49-year-old site is consistent with the intermediate disturbance hypothesis. For those that aren't familiar, it states that the diversity of species is generally higher in sites that have neither too much disturbance nor too little. And so that occurs because you have species that can thrive in both early successional stages and late successional stages and are coexisting in the same area. So some next steps that we have is we're going to finish our bite guide. So we're continuing to take more insects so we can expand that so you can see that if you come across those marks in the field, or more like to test at least maybe family or order, how close you can get. Right now we're only able to tell the difference basically between birds and standard arthropods, but hopefully you can get some arthropod diversity and see if that changes over the chronosequence as well. We plan to replicate the entire experiment, in addition to the fact that predation by natural enemies tends to vary day to day. We also had several major rainstorms during the 2 months that we were here, and because we were only doing a 2-day exposure, and we know that abiotic factors affect natural enemy behavior, it's possible that precipitation was playing a role, and we hope that by replicating the experiment, we address that potentially confounding factor. And then lastly, this study is part of a larger project that is investigating herbivory, vegetation, and plant community structure and seeding dynamics. So it's playing a core part of some ongoing projects in the lab as well. And I'd like to say a quick thank you to Anita, Maria, Marcelina, Amani, and Olimata and Liza for all of their help on this project, my university, my college for funding, and of course Stride, everyone at BCI. These past 2 months have been amazing, and I've loved having conversations over lunch about how I should keep the caterpillars on the leaves. So I really, really enjoyed that, and I'm super proud of this collaborative effort, and I'm excited to dive deeper into the results to see what we uncover. And with that, thank you so much for everyone for coming this evening, and I'd be happy to take any questions that you may have.